Hi everyone, in this video we focus on the development of B cells. Here's an overview of this B cell development. First of all, the B cell development starts in the very early fetal endogeny. It carries on in the late fetal life and it also continues after birth. The B cells that develop during the early fetal life originate from stem cells which are found in the fetal liver. However, these in the adult are just a minority, which is located in the secondary lymphoid tissues, and they are mostly involved in the innate immune response. On the other hand, B cells during late fetal life and after birth originate from stem cells which are found in the bone marrow, and they are the ones that carry out the adaptive immune response. And so we are focusing on these specific B cells. Here's the first stage of B cell development. We are now in the bone marrow. And in this first phase, the progenitor or B cell precursor, the progenitor B cells rearrange their immunoglobulin genes. So these are the immunoglobulin genes. Now, this phase is independent of antigen. There are no antigens present in the bone marrow, okay? But it depends on interactions with the bone marrow stromal cells. This first phase ends in an immature B cell that carries now an antigen receptor in the form of cell surface IgM. And now in this second phase that we have moved, it can now interact with antigens in its environment. What are these antigens? These are the self-antigens carried on the cell membrane of bone marrow cells, because we are still in the bone marrow and this is the environment so far of the B cells. Now in this phase, the immature B cells that are strongly stimulated by antigen either die or get inactivated. So these that react with the self-antigen undergo a process which is called negative selection. So in this way, these are removed. Many self-reactive now antigens, self-reactive B cells are removed from the repertoire. Now, in the third phase of development, the surviving immature B cells emerge into the peripheral lymphoid tissues and mature to express, except for the IgM, they now express IgD. B cells will migrate from the bone marrow to the secondary lymphoid organs primarily through the bloodstream, rather than the lymphatic vessels. They exit the blood and then enter the lymphoid tissues. The lymphatic network is more involved in the trafficking of lymphocytes from tissues back to the lymph nodes, but initial migration to secondary lymphoid organs is blood-borne. Once in the secondary lymphoid tissues, and now that they are mature and express both IgD and IgM, they can be activated by encounter with their specific foreign antigen. This happens in the peripheral lymphoid organs. Once they are activated, they proliferate and they differentiate into, first, antibody secreting plasma cells, and two, long-lived memory cells. To recap, the process of B-cell development starts very early in the fetus, carries on during the adult life with a continuous production of B cells from the bone marrow. Production takes place in the bone marrow. These immature B cells will migrate through blood circulation to secondary lymphoid organs, where they will encounter antigens and they will be activated still in the secondary lymphoid tissues.